Can you hear me well?
Yes, I can hear you really well.
Well, I think I'm going to hire you, Bhavna. I I need someone to introduce me like that to people. Uh, Thank you so much for the kind introduction. Um, yes, I know Bhavna for quite some time now from Moorfields days as patient, right? And then um, as the as the CEO of this amazing group. Is it the biggest Stargardt family support group worldwide, Bhavna?
I
Is
think
that where you stand right now?
I think it's the only one of the only
The
ones,
only one. Oh, right.
one of the only ones, yeah, in terms of like a formal organization with the work we do.
Mm,
We are
that's
global.
really good. So today I'll be talking a little bit about Stargardt disease. I always joke with with fellow scientists and physicians that this is the hardest kind of talk to do because when you're dealing with people in the area, scientists and doctors, it's all right. They may not even be paying attention to you, but you guys know this disease much better than me, so I can maybe add some things about research, but just to make sure we got everything covered. I discussed with Bob Miller for what we are going to be talking a little bit today. So I just thought, let me start by saying the Stargardt disease is really a journey of resilience. I think Stargardt disease is, as you may know, the most common form of inherited retinal degeneration. It's the second, it's actually the first and most common form of inherited macular degeneration, which is the center of the retina that's responsible for central vision, where we have the highest density of cones, which are the photoreceptors that give us um, ability to have stereopsia, color, good central vision, vision of detail, these kind of things. It affects around 1 to 10,000 people. So it for rare diseases, it's quite common. It's on the commonest side of the spectrum. Usually begins in childhood, as you know, but it can also emerge in adulthood. I think we may have some patients here. Some of you may have this start in adulthood, what we call adult onset Stargardt disease. Um, at the, I guess at the core of Stargardt disease, it's a storage problem within the eye, and we say that because the most common gene that causes Stargardt disease, right? The most common gene that has a mutation is named ABCA4. ABCA4 is a gene that's expressed in the epithelium of the retina, so it's like on the skin of the retina, below the retina, in the most external part of it. And what happens is that the process of vision it follows a very specific stepwise motion. So light comes inside the eyes, crosses the entire media of the eye, reaches the external site of the retina where the photoreceptors are, and then you have a cascade of events that turns the light into electrical impulses in the internal parts of the internal sections of the retina, which you have electrolytes coming in and out the cells, and this generates an electrical impulse that goes to the optic nerve, which is literally a straw that connects the brain with the eye, and the straw takes the information in the form of electrical impulses to the brain, so the brain can interpret that as an image. So I always say that the eye doesn't really see anything. It's the brain that sees. The problem is when you have areas that are not working in the eye, the image doesn't, or the electricity doesn't go correctly to the brain so that he can interpret it. And what happens with the ABCA4 protein, right, that's produced by the ABCA4 gene is that it works as a flippasi, as we call. So flippasi is a fancy word that only means flipping, really. It only flips what's inside the cells to what is outside the cells. So it's like an elevator, right? So it takes the buildup that gets hampered there after this entire visual cycle happens, which is multiple times in a minute, as you can imagine. Um, and then without this working elevator, this buildup of toxic material gets deposited in the retina. And I guess the most recognize of then is lipofuscin, right? Lipofuscin is a type of fatty byproduct, I guess is the best way to put it. And it gets deposited in the macula, which is this region in the retina that's responsible for central vision, color vision, sharp vision. Um, for many years, we didn't really have much to do, as you know. There's always the promise, right, for treatments, and I think the oldest patients in the group will know that there's always a talk about possibility of supplements and modified vitamins and that kind of thing. To be pretty honest, we learned much more about Stargardt disease after we got our gene sequencing capabilities in a better place. So now we know exactly what it is. Technology allowed us to see what it does, right? So we can estimate what it's doing inside the cells, which is necessary when you think of, we want to make a treatment in preclinical models, in mice models, and then bring this to humans. Um, so today is different because we actually have hope that some things are happening, and some of those are in late stages. As you know, I think Bhavin was telling me that you all had some conversations with Splice Bio, I think, with Belite Bio. So, these things are all coming. Some of those are in late stages, and I'll talk about this a little bit later, but just know that things are coming. Actually, they are really coming. We just don't know when that's going to be, but things are coming, and the pace that research is walking in Stargardt disease at least, and it's part because of the commonality of Stargardt disease, right? So there's plenty of interest from the biopharma companies to, build something to treat it. So it's very fast. And we're hoping that in the next more likely 5 to 10 years, we'll have some combination of treatments available. And again, we can never lose hope, right? We're talking about Rare Disease Day. We can never lose hope, but it's coming. We just need to, I guess, this time we actually need to be a bit patient. Um, so I think this landscape has changed. We are in a process where we innovate. Now we have on not only these oral medications, we also have gene therapy, and gene therapy has a funny story. I would say kind of bumpy story in Stargardt disease because ABCA4 is a big gene, and I know that there are other genes that can also cause Stargardt disease, right? ELOVL4, which causes an autosomal dominant form of the disease, and then we also have PROM1, but the most common one by far is ABCA4, and I guess it's where the focus in treatment is right now. So gene therapy, we could never really do gene therapy for ABCA4, and that's because uh, the way that you do gene therapy is not only releasing a medication for the eye. That's not how it works. You actually need to put the sequence of the DNA that you want to have an effect inside the nucleus of the cell, so that can actually become the new blueprint that the cell is using um, to work. So ABCA4 is a big gene. The vector, which is the vessel that we use, is an adeno-associated vector. Is a virus, right? It has like a third of the capability to put an ABCA4 gene over there. So ABCA4 is much larger than a traditional adeno-associated viral vector, what we call AAV. But now we actually have some very elegant colleagues of ours who are doing some, very, very cool things with this. And actually, we have dual vectors now. So things are walking in a fast speed. And what is, I guess, most relevant of this is that it's not only our knowledge of Stargardt disease that's getting better. So it's not only that's why I always tell patients, if you have if you're eligible for an observational study, perhaps participate, because something's coming out of that. It's not only to get funding. Some people think that scientists only do natural history studies to apply for funding, but that's not how it works, right? We learn so much more of Stargardt disease with things like ProgStar, the natural history study that we have in the US, the natural history study that we had in Moorfields as well. So we learned a lot from these things, and it allowed us to come up with all of these different things. And then we also have groundbreaking tech, literally groundbreaking tech. Um, Optogenetics, stem cell um, is also going on at the same time, and we are very very excited to see these things rewiring the retina. Um, and then I think um the before we get to this part, I just wanted to let you know that we are working as hard as we can to make sure that things happen. And it's not easy, as you know, but we're trying our best. So we have top people working on actual treatments, which I think is the focus of today. Um, anyway, in terms of some of these things you have already heard, so I'm just going to honestly just go through it as quickly as I can. Uh, One of the first, I guess, approaches in terms of supplement was the ALK 001 study, right? The Gildo retinol from Alkeus. It was one of the leading candidates in development for quite some time. It's a form of adulterated vitamin A, and by adulterated, it just means that it's an isoform of vitamin A that's modified to be a little bit more stable, and in this way, it reduces the dimerization or the catalyzation that generates these toxic products that get accumulated in the epithelium, right, in the skin of the retina, and causes the damage that we see in Stargardt disease. This one has a long story, right? We had T's one, we had T's two, we had T's three, and then the FDA asked for a new phase three, and that's where we are right now. We The last updates that came from and I just have this written down here because I don't really remember. We had some updates on the end of 2025, and they what they said is that over 24 months, and again, this is all commercial information, right? So most of these actually have not been published yet, so we don't really know. But over 24 months, it was well tolerated. The treated participants had a preserved retinal structure and function, um, but we are just looking forward to hearing where they are with this because it's been some time in the making, and we hope that this actually goes ahead. Just to give you an idea, in the phase two trial, there was a decrease in 20%, right, of the atrophic lesion progression. And the atrophic lesion is where you have the thinning in Stargardt disease in the very central macula. So we are excited, right, because it's not a cure, it's not a definite cure, but it's a way to stop or to hamper the progression of the disease. And that's why I think it's a combination of treatments in the future, right? Is using some sort of supplement to reduce the progression with something else that may improve function or that may replace the cells completely. Then we have, sorry, I should have said that. I don't really have conflicts of interest, but I worked in this study that I'm going to talk right now, right, in the Dragon study. But that's it. I don't have any sorts of financial disclosures here. So the next study is from the Light Bio. I think you guys heard from them. They're testing tinlarebant, which is a medication that's being used for age-related macular degeneration, which is another form of retinal condition that's non-hereditary, or at least not completely hereditary, and then also Stargardt disease. It's a retinal binding protein 4, and what it does is just, in simple terms, it just reduces the vitamin A that's available for the retina, right? And then in reducing the amounts of vitamin A, you also reduce the amount of toxic byproducts that get deposited in the epithelium of the retina. The Dragon study, as you may have heard this quite a lot these days, Dragon and Dragon 2, um, had very good safety profile and also some efficacy in the early stages. And then the the lesion growth in the participants was reduced by approximately 50%, but this is before they actually gave us any efficacy results, and we actually had later 2025, I think it was October, the actual final results that they're submitting to FDA and asking for a new drug approval, right? They're filling the form to request this to be actually usable, so people can actually be prescribed this. They had a 26%, if I recall correctly, a reduction in the toxic byproduct accumulation, and this was measured in the way that the in the way that we do the test, so we can actually measure the size of the atrophic lesion. And again, that's the importance of natural history studies, right? We know from natural history studies in which speed this atrophic lesion progresses. And what happens is the reduction was quite remarkable, and I don't really remember now if it was 26 or 36%, but it was quite a decrease. And this was significantly using statistics as well. And this is remarkable because it's better reduction than the Alkus project, for example, and also it's something that can be used in all age groups. So we're very excited. We are hoping that they get this drug to the FDA. The plans, I think, was submitting a new drug approval form um, in the first semester of 2026. Of course, these kind of things, because of stakeholders and other stuff, they don't really share with everyone. But that's the plan where it currently stands based on their last, and I have the website open here in front of me, based on their last commercial update last year. So hopefully we'll have something very soon, right? Because the FDA they have a deadline, right, to respond to this, and they ask that the biopharma company to, in case you're not aware of how this works, they ask for the biopharma company to send some further information, and then it's a process of a few months. So we hope that this becomes something that we can actually use. Then we also have we had another study, right, which was terminated, emixustat, which is a visual cycle modulator. I don't think you will have heard from the sponsors of this, which is Kubota Vision. It's an oral molecule that inhibits the RPE65, which is a protein also involved in the retinal pigmented epithelium, and it's involved in the visual cycle as well. Actually, there's also even a gene therapy for this specific gene, RPE65, but it's for an entirely different disease, Leber congenital amaurosis, and it's the only gene therapy actually approved for an eye disease. And what it does is that by acting in what the RP65 is doing, it reduces the product of the production of the visual chromophore, uh, thus also limiting the buildup of toxic byproducts in the retina. The I don't really remember when we heard from them, but the name of the trial was Sea Star. It's been quite some time, I think. There was some promise in the early studies, but the phase three terminated because they didn't really meet the primary endpoint, which always in Stargardt is going to be the reduction of the area of atrophy in the macula, and because they didn't reach that, the study was terminated by the sponsors, which is how it usually goes when things don't really show the effect that they're looking for. And then we also have metformin, right? And this is led by one of our great friends, Brian from the NEI, National Eye Institute in the US. They're testing metformin, right? Metformin is an interesting one because um, we we didn't really know. When I first heard about metformin being used for Stargardt disease, I was surprised because, as you may know, some of you may have diabetes, right? And metformin, or you may live with someone who has diabetes. Metformin is a disease is the most common actually drug for diabetes. And then they realized that in animal models, there's some lipid defects, right? there was lipid accumulation in the photoreceptor outer segment. So they decided to test metformin in adults actually to see if targeting these lipid metabolism defects could actually enhance patients' functions and also reduce the rate of progression of the disease. Um, Preclinical evidence, uh, so comes from mice models in this case, was promising, and they're testing this in 50, I think it's 56 patients with Stargardt disease in the National Eye Institute, and we don't really have final results yet. We are currently in the area of 24-month follow-up period. So hopefully we'll hear something soon, and it's likely to be published as well. So that's another possible approach, right? Which would be interesting to have metformin, which is a medication that's already available for another very common disease to be used for Stargardt disease. So it's a very nice approach that doesn't exactly target the mechanism that's causing Stargardt disease, but it targets something else that also contributes to the condition. So is it possible that in the future we'll have something like, I don't know, tinlarebant to target vitamin A and also metformin to target lipids? I'm not exactly sure. I don't think anyone has that answer yet. But that's the current landscape of where we think we are going in the future. And then we also have the gene therapy approaches, right? And I think this is the part that you may have heard a few things from Splice Bio and from other companies. I'm just going to touch on a few of these in case you're not really uh, uh, well versed in this. So the landscape for the most advanced forms of therapy for Stargardt disease uh, is very promising. We have highly innovative and technologies that truly bypass the photoreceptors, and we also have gene therapy, which is used for other forms of inherited retinal disease. And what's I always say this to my patients. We live in the era of genetics, right? So gene therapy is actually coming for a
Tienes
variety
razón tú.
of inherited retinal diseases. Sorry? Yeah, you can ask. Go ahead. Is there a question there? Hello?
I think we can go for questions afterwards. I think they might have just joined, maybe. And
Oh, okay. Sorry, I can't see you guys because I have myself a small version of myself here in the bottom,
no, that's fine.
and I'm with the last updates that they gave open here in front of me. I would
No,
just
that's
stop me at
fine.
any time. And then so we remember about the size of the ABCA4 protein, right? So we talked in the beginning for those who just joined, uh, how gene therapy has had a funny story, right? With a bumpy road with ABCA4 and gene therapy, right? So the issue is the ABCA4 gene, which is the most common gene to cause Stargardt disease, is a very large gene, right? So I would say the type of vector that carries this inside the cells that we're most used to using is AAV, which is in short for adeno-associated viral vectors. And this essentially is a small body of a virus, like the viruses that cause common cold, for example, common flu. So you just empty that using bioengineer and you put the sequence of the gene inside this vector so you can actually deliver the genetic material, the protein, whatever is it that you're delivering inside the nucleus of the cells. The issue being that ABCA4 is quite a large gene, and so it's triple the size uh, of the most common use viral vectors, close to triple the size. So the first attempt in navigating through this was using a lentiviral vector. So a lentiviral vector is a genetically engineered replication deficient virus, right? Derived from the HIV-1, which is the immunodeficiency uh, uh, virus, right? And the thing about the lentiviral vector is that it's quite a big it's quite a big sorry, they're doing some work outside here. So it's quite a bit of it's quite a big vector. It has a big capability, and it also has been used for other diseases like age-related macular degeneration. We ha actually had gene therapy for that condition using lentiviral vectors. The issue about lentiviral vectors is that it has been shown sometimes in some studies that it doesn't really have the expression capability, right? The transfection capability of the adeno-associated viral vector, which just means that it doesn't express the gene as well as the adeno-associated viral vectors. So this is one of the limitations, but because of the size of the gene, we attempted to do this with lentiviral vectors. And the first study represents one of the first direct gene supplementation approaches for Stargardt. It was a study by uh, Sanofi. I'm not sure how you say that in English. In Portuguese, it would be Sanofi or Sanofi, I guess. I don't know. I don't really know. But it was a phase one, two-way study with around 20 participants. And when we say one slash two-way, what that means is that it's a safety study, right? Seeing if this is actually safe to be used in humans. And then the two-way phase is when you escalate the dose and you try to find which dose is the best to treat the patients. So it was a safety slash a dose escalation study to investigate this product, which was called SAR 422459. It's quite the name. It was well tolerated. The adverse events were related to surgical administration. This was actually many years ago now. And as you may know, subretinal delivery, which is where this gets delivered close to the epithelium of the retina, involves a surgery called vitrectomy. Maybe some of you have already heard of that. A vitrectomy is essentially a word meaning to empty the globe, right? The jelly inside the globe. So we have this jelly material. If you touch your eyes, you will feel. That's the vitreous jelly. So this vitreous, you need to remove that so that you can actually reach the retina and inject the liquid inside the retina in this external part. This causes, of course, a retina detachment, but we know it's safe. We do this for plenty of treatments and actually plenty of genes. And actually, the gene therapy that was approved is exactly doing this. It's a vitrectomy followed by a subretinal injection, and we know it's safe. Um, no, the issue with this study, it's not really the safety. It's just that there were no clinically significant changes. So there was no change in visual function. And I think a third of the patients that were treated also showed a bit of a progression in the atrophic lesions. Um, so it's just it didn't really show any signs of efficacy, although this is a safety study. So it didn't really go forward as far as we are aware, or as far as the sponsors have said. We have the long-term study currently ongoing, and I think it's up to 2030 or 2033, I guess. So it's still a few years so that we know how this is going to show in the long term. And this is very important because we had some treatments which, you know, in early phases they were good, and then when you do a phase three and you do the extension, the follow-up study, there were um, no big efficacy signs. So it's important to have follow-up studies as well, not only for safety reasons, right, but also for measuring efficacy in the long term, serially speaking. Um, and then we have this, and this is where I say it's very elegant. We have Splice Bio, which you have already uh, heard from them. There we have some new vector technology being developed, which includes dual AV vectors, AAV being again the adeno-associated viral vectors. And the elegant part about this is that it splits, right, the gene into two coding sequences so that it can actually uh, join later. And what it's doing actually, if you think of the gene as a series of instructions, of a strand of instructions, what it's doing actually is that it's getting this strand, cutting it in the middle, putting a molecule here on this side and a molecule here on the other side. And what it does is after this gets inside the cells, these molecules, which we call split inteins, which is just two part proteins that join gene regions, they join, right? And when they join, they reconstitute the full length of the ABC4 protein. How cool is that, right? That's how far we've gotten in research. And the preclinical studies showed a great reduction, first efficient reconstitution, right? Because you gotta know if it's actually joining, right? Otherwise, you have broken parts, broken segments of the gene that doesn't really work. So it does. It showed efficient reconstitution and also it showed that a reduction in the bisretinoids, which again is the toxic waste that gets deposited in the retina, like lipofuscin and these kind of things. So these findings led to the first human clinical trial. I think they actually already dosed the first patient. I'm not sure because it depends on where you look, actually. Some places say that it's currently enrolling, which we know it is. The sponsors haven't actually said that they dosed the first, not at least an official update. But if you go to Foundation Fighting Blindness, which is another um, great research and family support groups for patients with rare disease, they actually say that they dose the first patient. Now, I'm not sure if this comes from, and some sort of announcement that wasn't really published anywhere else. They do some podcasts, so I'm just guessing, maybe one of the medical monitors or one of the key investigators gave some sort of announcement there. What's important is that it's ongoing. It's a phase 1/2. So again, it's a safety phase followed by a dose escalation phase, which they escalate 2 or 3 doses to find which is the optimal dose. And then they go for the expansion phase. So literally, they find the optimal dose and they extend that dose to the other patients as well. And what follows this is a phase 3 study, right? Um, the FDA has been recommending actually some studies to have a phase 1 and 2 that are placebo controlled, which is interesting because we had some other studies from ProQR, for example, that showed a lot of signs of efficacy, but they didn't really go through. And then Daniel Chang and people who are involved um, in these studies as medical monitors or as medical directors of these companies, they actually had some discussions with FDA, and they are actually recommending some placebo-controlled phase 1 and 2 studies. So there might be some changes in what we're seeing in terms of design of these studies moving forward, which is interesting. Um, but we never know, right? It all depends on the sponsors, and they always communicate with the FDA and the regulatory agencies. So let's see how that goes. Um, and we're very excited, right? Because this approach it's again, it's very elegant, right? It's very refined, and it just shows us how far science has come, I guess. And then we have some gene therapy, gene modifier therapies, right? So Ocugen has a project named OCU 410ST. It's quite the name, right? So when, just to clarify what this is, when it's on a early phase trial, they actually just put a big name just for, they have their own kind of naming inside. And then when it goes to a phase 3, there's some promise that this will go forward. They actually have to name the medication. So this, the one for, I actually had to look as I don't remember, the one from Sanofi, right? star 422459. If this went ahead, this was going to be something else completely when it gets to the phase three. So this is a preliminary kind of name. But what is interesting about this is that it's not actually treating ABCA4, right? It's not actually targeting ABCA4. It's not replacing that protein. It delivers another gene named RORA, which is it's an isoform of RORA with an adeno-associated viral vector to regulate the homeostasis of the retina, right? And the mechanism that it does this is by targeting inflammation, oxidative stress, lipid metabolism, a complement cascade, which are things that have been linked in the past with Stargardt disease or with the damage that causes Stargardt disease. And again, that's the importance of natural history studies, right? We learn many of these things with natural history studies, with animal models. So never underestimate the the power that this can do for research. Um, and this was it's an open label phase one slash two dose escalation study. They only targeted, I think it's 10 or 9 participants, and again it's a subretinal injection. And the subretinal injection is again followed. Um, it's after a vitrectomy, which is the surgery where you empty the jelly material inside the eye. It was tolerated. After 6 months, the eyes have shown some sort of improved visual acuity and then also stable lesions with the atrophic growth. And this is where it gets interesting, being 54% slower than in the untreated eyes, which is quite remarkable, right? We're talking about reducing the natural history by half, right? Hampering the growth of this. We're not exactly sure yet how this translates to vision, right? Because remember that one thing is what we show in the structure, the other thing is what we show in the vision, in the function. And sometimes these don't correlate well, but it's remarkable, right? It's showing us that actually they were able to reduce the disease in the treated eyes by half. One problem with, I guess the last 10 years, there was a lot of changes in the FDA, particularly, where you can't really compare anymore, right? Treated eye with untreated eye. We used to do that. We actually have to do this with placebo control groups. So, we can only show that, okay, you actually reduce the progression in this eye um, than compared to the eye that was not treated. But for the purposes of of regulatory bodies, this doesn't really go that way. So they actually need to go to a phase two, which actually they are going ahead with that. It was recently approved, um, the phase two entry by the FDA to start. So that's great. It's a gene it's what we call a gene agnostic strategy, which it's a word that I love because all of these therapies, if you think about what they are, these are actually gene targeted therapies, right? So we're talking about targeting ABCA4, targeting another gene, targeting RPE65. But what about PROM1, for example, right? What about the other gene that causes Stargardt disease as well? And this gene agnostic strategy offers a promising alternative, right, to conventional gene therapy because if this goes ahead, then it's targeting the mechanism, right, that causes inflammation, the cascade complement, the complement cascade to be activated, lipid metabolism. This means that you can actually use that for other things, right? And there's another very interesting study. I was talking about Daniel, right? Daniel is involved in this specific study, which the name is Prodigy. I forgot the name of the farm actually, but I'm forgetting a lot of things. I'm sorry about that. I have 3 babies, so my life is crazy. I was telling Bhavna before I started that I had plans to do some slides, right, for some of you to follow. But my babies had another plan entirely, so I wasn't really able to do anything. Uh, but I was able only to prepare to this, right? The other therapy is named Prodigy, which is also a gene agnostic strategy. It wouldn't really be effective for Stargardt disease, but what it does is actually it it gives the cells of the retina a specific molecule that the rods, which is a type of photoreceptor that we have. We have cones and rods produced to keep the cones alive. So when you put this specific molecule that it produces inside the viral vector and you deliver this to the retina, what happens is you're delivering a protein that keeps the cones working, right? And that's the thing in rod cone dystrophies, for example. And I think it's not exactly what we're doing here, but just so you know, I know some of you want to know about things that are happening that may also be used for Stargardt. The cool thing is that you know you're actually you can use this for any sort of disease we think, right, that affects the rod and the cone system. So rod cone dystrophies and these kind of things. So it's pretty cool. We don't really know where this is going in the future, but it's good to know that it's not only gene targeted deliveries. You also have gene agnostic products coming. Let me just speed this up a bit because I lost a bit of time. We also had RNA editing, which is a different target, right? If you think of how it works, you have the DNA, which is a blueprint of what we are, right? So you have the blueprints of the ABCA4 gene, for example, which is the DNA sequence of it. It gets transcribed into an RNA, and then this gets translated into a protein right inside the nucleus of the cells. So we have some targets, some products that are targeting directly this level in the transcription level, right, in the RNA. And Assyrian Therapeutics, they already started the study. We are eagerly waiting for the preliminary data on its safety and efficacy. The name of the study is Stellar. It's a phase 1/2 study, and again, RNA editing is very promising. We think this is going to help us target a lot of the diseases that we can't really do gene therapy on. So it's very, very cool. We hope that the it's safe and the efficacy is actually shown. I think they're going to, give some updates soon in the first semester of 2026, because they are due to provide some updates. And then we have optogenetics, right? And I'm just speeding this up a little bit. I'm sorry. It's a very elegant approach. Again, it introduces photosensitive proteins into retina cells that don't normally respond to light, right? So remember that for the vision to work, right, for us to see something, the light has to go in the external side of the retina, and this gets the photoreceptors get to translate this into an electrical impulse that goes to the brain, right, via the optic nerve. But for this to happen, it has to do a cascade of events that sends the information to the inner sections of the retina, like the ganglion cells and the bipolar cells. And then this will turn into an electrical impulse that goes finally to the brain. So the plan here is to actually target those cells so that we can bypass, right, the photoreceptors that are not working properly. So it's very cool. And again, this is gene agnostic, right? It doesn't require gene testing. It doesn't require major surgery. It's done by an injection inside the eye. It doesn't require external devices, right, like goggles. So it's pretty cool. The name of the study is Starlight. It's the phase 2A actually was well tolerated. Um, to be that honest, we were very surprised with the results because we're talking about 3 to 4 lines of visual improvement, right? And, and of course, we need to um, keep our feet in the ground, as we say here in Brazil, just because we're not sure, right? Until we see the final results. But the last update was in Italy. It was in the Flow Retina Congress. I think it was in December of last year. And Alan Ho, which is one of the physicians that is involved in this, I think he's the chief investigator actually. He's from Moorfields Eye Hospital in the US. And they actually shown that the optogenetics project that they're testing, the MCO010, restore vision, and this is this is not how do you say, it's something that has been the effect is something that is maintainable. It's not something that you had a small effect in the beginning and then you don't have that shown in the long term. They're actually following the patients now for over 48 weeks, which is almost a year, right? And it's very exciting. We hope this is going to be one of the gene agnostic products, right? So it's very, very nice. We are hoping, we're wishing the best of luck to them in this study and, Godspeed, as we say, because this is truly remarkable. Let's see how this goes. They are in the phase 2/3 now, so we're going to hear the final results very, very short. And then just to finalize this, right, we also have stem cell therapy. It's quite a lot. Uh, I'm Brazilian, in case you don't know, we talk a lot and we talk a bit fast, like the Spanish when they are talking to each other. Uh, and what's funny is that I'm speaking in English, but the verge to speak hasn't stopped, which is quite remarkable. I think I should put myself in a CT scan to see what's going on in my head right now. But then we also have stem cell therapy, right? And the first one that we used was derived from embryonic stem cells. This was a study actually in Moorfields a few years ago. This type of cell, right, that you can derive stem cell from quite a few cellular types: bone marrow, induced pluripotent stem cell, human embryonic stem cell, and we use embryonic stem cells in the beginning, derived from the RPE, which have shown some effects, some good effects in preclinical models again. And there was a very early trial by Stellus, which is this one that I'm talking about, showed visual improvement in 4 out of 10 participants. I don't think they're going ahead with that because they had some side effects, which were related to the treatment, not to the medication, but to the way that this is done, because you actually need to put that in after a big surgery. Sorry, these are my kids. And then you also have a new therapy by Blue Rock Therapeutics, which started, the name is OPCT-001, and they are using induced pluripotent stem cells. We think this is a much more promising cellular type, actually, than embryonic stem cells or bone marrow stem cells. And there's also the last update that we heard is that there was some bone marrow studies derived stem cells, named Scots and Scots 2, that are based in the US, that have shown a functional improvement in 61% of the eyes. But again, this has not been published. We are looking forward to hearing the final results. This is a study that's going on until 2027. So this is, I think, where we are treatment-wise. I try to do something just to put everyone up to speed to everything that's going on. So innovation in Stargardt, as you can see, is moving very fast, as we kind of touched in the beginning of this. In the near future, I expect we'll see a combination of treatments, right? And many patients ask me what's my honest expectation of the future, and I'm a very optimistic person, but I believe, with all sincerity, that in the next 5 to 10 years, this will be transformative for Stargardt, where we're talking about possibly oral supplements reducing the progression of the disease, and then also some form of treatments that directly target the retina or the photoreceptors or bypass the photoreceptors to actually improve function as well. So restore vision, right? Because it's a funny one, because for the physician, sometimes as scientists we say, oh, actually there's a reduction in the progression of the disease, but what exactly does that mean to the patient? Did the vision get better, right? And that's one of the things that we're trying um, to circumvent. So hopefully, we have some very good results soon and some new updates. Hopefully, with new medications being used for patients. Right. In the meantime, what we tell patients is, keep a healthy diet, eat plenty of green vegetables, dark green vegetables, fish as well, particularly fat fish. Use sunglasses with good UVA, UVB protection when you go outside in the sun, and don't smoke. These are the things that we know that have been associated with a reduction um, in some damages in the retina from other conditions, as well as Stargardt. So it's just something to keep in mind while we don't get to the more, I would say, specific treatments. Thank you so much for your time and for, listening to me for almost 1 hour now.